I think customary at this point, I apologize for giving an incomplete summary of what people talked about, but just to sort of indicate a few connecting threads that I think we can pick up on for the discussion here. We don't have all of our speakers here, but on the call, Hanna, can you just confirm you're with us?
Yes, yes, I'm
Yeah, you can share your screen as well if you like, so we can see you. If you don't want to, I suppose that's okay. So we have Hanna. So Hanna, David, Raphael, myself. Again, sort of brief summary. So amongst other things, the work I was telling you about this morning was about, at the end of the day, brain networks. There's actually an element of multi-scale spatial that I might actually sort of bring up here that wasn't featured much, but is actually quite an important part in the analysis of brain imaging data, and yeah, covariant structure and network analysis. Rodrigo was Raphael was telling us about his work on time sequences, and how those are potentially encoded in sort of these Poisson rate models. Chrystopher told us a whole load of things about algorithmic complexity in agents and things that were, I think, new to me and maybe some other people in the room. Hanna, we just heard from most recently. So I think some of the connecting threads here are time, and it was great, Chrystopher, that you had an extended kind of set of general ideas about time. So I think we'll come back there. Time, sequences, scales, some things in that direction, dynamics as well. But I want to start actually between Hanna and Chrystopher. So just initially, what's the connection here? So how did the project that you're working on, Hanna, sort of evolve out of what you've been thinking about in your own work, Chrystopher? So I see elements of the connection, but I suspect there's a lot more in the background. So yeah, maybe you could fill us in on that.
Yes, so this epsilon machines that I've considered here in terms of the application for the EEG microstate sequences analysis, they are themselves constructed from the data because we have in the structure the event-driven transitions, we have the transformations of the state space, so we have a corresponding semigroup for every machine. And we can look at all of those permutation structures inside these models, which can give us like additional insights on the structure. Because here we mainly we use this distance metrics, which is like L1 distance on the probability distributions, which also takes into account how the words were clustered. But what we can also do is look at the other properties of the machines, and particularly the transformations and the semigroups and the complexity and different ways, different algorithms of constructing these machines from the data can allow us different, like for some of them, for example, give more of a periodic structures where we can't find much of a permutations, and the complexity tends to be like this measure, particularly Kronos complexity, to be one all the time. But if we want to look more for the periodicities, for the permutations, and maybe find what also then like going with this application to the EEG data, what patterns are like periodically repeating, what classes of words we are going around. So this can be extended into this sort of analysis that Chrystopher told about for these models in practice.
How did you do, Chrystopher?
Yeah, nice.
I think there is no sound.
Sorry, I mentioned a lot of the automata that we had. I think as an artifact of the construction method, you get low Kronos complexity, but there's another complexity measure for aperiodic automata that don't have groups, which also gives you complexity measure. Actually, Hanna's done published work on, not applied to EEG, but just on giving coordinates on such systems and computing their complexity. So that could help. But your question was about so where the different time scales come from, and I think they're very natural because you have these different ways of looking at the data. Because if you're just looking at a time series of recorded EEG, you see all these lines. And then because of the clustering that you referred to, k-means clustering, and people have different methods to do it, you get a discrete alphabet out of it. That's small discrete alphabet of size 4, 5, or 6 usually, and that gives you. So every moment of time, you can see what letter you're at. That's the clock sequence, and it is, as you said, it's downsampled. I think it's recorded 1,000 megahertz and downsampled to 250. But that's clock time, and clock time is one granularity of time that you can look at. But if you look at actually a lot of the software that does EEG microstates, they interpolate they interpolate over just using the peaks in the global field power, as Hanna showed in the formula. And so actually the measurements that really matter are just at these global field power peaks, and they happen not at a regular clock time exactly, but about 50 milliseconds with some variance, right? So that's a natural clock in the brain, if you like. Which is not, it doesn't actually exactly match up with our clock time, but it's a kind of event that's happening regularly in EEG. So it makes sense to use that as the clock tick for that scale. So that's the peak scale that she was referring to. And then if you're just looking at, okay, I want to know if this microstate, like in the schizophrenia, there was the work by Lehman and all detecting schizophrenia by certain EEG microstate sequences. All they looked at is which microstate is followed by which one, ignoring the clock, don't care about Newtonian time, and ignoring the peak time also. So they're not looking exactly when it happened and how many times it happened maybe it lasted 300 milliseconds and this one lasted 60, the B after it, but just collapsing it down into the change of microstate. Right, so that's a natural way to look at EEG microstate data. It's just when do you have a new letter, and we call that event time. And so those are 3 different temporal scales for looking at EEG microstates, and then you're getting different kinds of separation results depending on where you're looking. We're also, and because you have a metric on the epsilon machines, you've got a metric that is a metric that is distinguishing different dynamical systems. So it's not just syntax. Syntax is there too, because it's in those causal states. As she showed, these are the characteristics of these causal states in the automata that accounted for the most difference between the groups or between the mind modes. And we've got some kind then interpretation of we've got a syntactic marker of something that seems to be functionally significant, but then we would have to go to the experts on, microstate D that kept occurring from mind wandering. That's not surprising because that's associated with default mode network, right, in the mind wandering state. So, and there were certain kinds of syntactic markers of that in the analysis. There's another thing I wanted to mention, but I'm going to come back and give somebody else a chance to talk.
That's great.
I know it. Yeah. Oh, and there's another direction you can take this in, because you could do, as Hanna mentioned, you can do this kind of construction of epsilon machines if you've got any kind of discrete alphabet. So if you take not just EEG, but if you take fMRI, you can go to another time scale where you can do k-means clustering and then try to put fMRI activation patterns into clusters, so analogs of EEG microstates, and say. So we actually, David, in the UK, one of my students, David, has looked at this a bit in his PhD, didn't end up working on it, but he looked at simultaneous EEG fMRI data, discretized the fMRI, and then you could do the same thing. You'd say, well, every TR, so every about 1 or 2 seconds, depending on your scanning time, you get a letter. right? And then you can say, okay, I don't care if I get the same letter. I only want to look when it changes. So you also get the clock and event timescales there, but it's much different. It's 2 seconds and above, maybe 16 seconds or something, depending on how you do it. He didn't end up doing that as a major part of the PhD he did, but he did look at the possibility of trying to relate those different levels, those 5 timescales. So 2 timescales for fMRI and 3 for EEG. And one can imagine like a hierarchy of epsilon machines describing each level and how they're related.
So when you're doing sort of planning these projects and thinking about this this application of the epsilon machine, symbolic approach, what are the reference points? Like, what's the success stories of that as an informative data analysis tool in some other area of, let's say, experimental science? Are there, I see how you could say, well, EEG data is kind of handy, like has the right properties, let's do it. But probably there's been some point over the last few decades, some really useful, informative, and I don't know, field transforming use of this, but at least something that was deemed like that worked out. Okay, let's use this in other areas, or let's try and sort of get the level of added value from a mathematically complicated analysis that we got over here in the EEG. Is there like a reference loosely in your head, like where that's worked in other fields?
So maybe Daniel may know more about this, but that's direction initiated by James Crutchfield. Now he was at Berkeley and is now at UC Davis. I think it was Santa Cruz in the meantime, and he comes from physics, and he's applied this to describing physical systems, and so it actually comes from physics. Daniel, do you know the killer app for epsilon machines from Crutchfield? So the field's called computational mechanics.
The main problem is that it is strict. Epsilon machines I think epsilon machines, the problem that they face is that they are strict. There's a information bottleneck way to express them, and information bottlenecks have strict bottlenecks, so they perfectly preserve all the information that's preserved. That's what the epsilon machines are. And then there's the soft version, which is much more forgiving, and where you can basically zoom out and make it softer. And in my opinion, that's what you would have to do to make it practically useful. I don't know. People have used it for various things, especially EEG. I do think EEG is a particularly difficult one because there are a lot of extra structures. So I think here zooming in, zooming out might be actually something worth having. So I'm a bit skeptical but I should be lying if I say, oh, there's none. Probably there are, but I'm not aware of the application.
Okay. I liked how you mentioned zooming in, zooming out, because that was something I wanted to move into. So thinking about kind of concepts of multi-scale on time dimension, right? Coarse graining and also just system organization. Where again, I'm thinking, are there good reference examples of where there's something interesting in any field of your choice, but particularly for like multi-scale? So in most of the cases that I think I'm familiar with, say within modality in neuroscience, I mean, we have multiple modalities, as you mentioned. We have, I say, we're going from imaging down. We have fMRI, we have EEG, we have a lot of things in basically those two time scales, like seconds and then milliseconds and sort of tens of milliseconds. And we don't typically try to go down a lot further, but we might be interested in some sub-millisecond stuff in at sort of membrane ion flux kind of level measurements. So there's sort of two big time scales. Then you can go slower. Yeah, like so there's some slow variations in say bold signal, tens of seconds. So what like two orders of magnitude, 3 orders of magnitude there, but really like two anchoring temporal scales, maybe driven to some extent by methodologies, but maybe just characteristic temporal scales. So again, in other areas where you might have some interesting observations, do we get many more than 2 characteristic scales? Do we get 5 characteristic temporal scales that are all qualitatively different, that we sort of measure with similar things, so they're not just completely different domains of physical measurement, for example, or physical theory, we think of as the same system, but it's completely different over here than it is over here, and it's like very richly multi-scale. I'm wondering if you have any thoughts on when you've seen that, if you've seen that.
Well, I see it in analysis of climate and climate prediction models. You have to do. I'm not an expert in this area at all, but you have to model what's going on in the ocean and in the atmosphere, and these things are operating sort of at different time scales, and then vortices in the atmosphere, and there's a big computational problem to integrate all the data necessary for that and then try to make predictions. But you've got this and for some of it you need to go very fine-grained locally, and for other parts of it you don't. And I'm not sure how many scales, but probably 5 of them are important in analyzing climate and the flows of all of these fluids over the Earth. You've got different types of fluids, right? Water and air are different in their properties, and you've got to integrate the slower and faster timescales. I recommend, there's a survey book on complex systems, Introduction to Complex Systems by Paul Figut, and he kind of surveys, it describes those things for people who are new, like me, and to that area. So I think that's one. And of course, the brain and behavior, human behavior at multiple timescales, and as you went from all those different levels, so kind of event-driven human action versus moment-to-moment awareness.
Right. Yeah, I'm probably cutting it off a bit short if I just say seconds, tens of seconds. There's lots of, minutes, hours, years, aspects of the brain and the mind and the body. But I do feel like those are like the main 2 anchors, like millisecond, second.
The whole temporal horizon of storytelling, history, philosophies, political movements, and relating that down to what your neurons are doing.
Oh, political movements, well. Raphael, thoughts on timescales?
I just wanted to mention, not that absolutely rapidly, there's somebody who knows more about that.
No, I was going to bring up a paper which may be relevant to some of this discussion. And then at some point, I realized that one of the co-authors is actually sitting behind me. Anyway, so it's a paper from a couple of years ago by Fernando Rosas and a bunch of other people, including Daniel. I think the gist of the paper is looking at emergence in the sense, in this case, of what they call computational closure. So it's looking at coarse-grained processes and comparing the epsilon machine for the full process and then the coarse-grained process. I'm not sure if it's particularly practical. It's probably not particularly practical, but it's sort of interest as to what you can do with epsilon machines, maybe. And one interesting thing I think they show is that, in fact, computational closure, I'm not sure if it's always or sometimes, turns out to be equivalent to informational closure in the same sense that I use it in my own dynamical independence thing. Again, I'm not sure. There are a bunch of worked examples in that paper, including, I think, the Ising model, a few other things, which does sort of seem to imply it can be used.
Some examples for the EEG, but not in that paper. That's why.
But not EEG. But I wonder if that might relate to, things like symbolic transfer entropy, for instance, might fall into that sort of bucket in some way. I'm not sure, which is a technique I've always been a little bit skeptical about. It sort of tends to sound better on paper than it does in practice, or something like that. Anyway, can I just also make a brief point just about timescales? Is that so? Certainly in statistical physics, timescales are kind of key. You have slow variables and fast variables, and fluctuations happen at one timescale, and so and that seems to fall naturally out of statistical physics. Why should it not fall out of complex neural systems as well? I don't know. I don't have an answer for that. I just wondered if you have any thoughts on that.
So I think it's more a question of how we analyze and think about it than whether it's there. Like whether it's there and how we can find and learn about it is one thing. And then there's sort of what's easiest to base your measurement scales around. And yeah, these seem to be confounded aspects of the practical process of scientific discovery. Like we can measure fast things, we can measure slow things, we can measure large things slowly, or large things at coarse grain. We can measure small things at fine grain, but we can't do kind of all of the permutations.
I mean, maybe in neural data, the fact that it's kind of 1 over f implies that there's stuff happening at all time scales, and yet there also seems to be particular stuff happening at particular time scales.
Yeah, I've slowly over the years come to the view that almost anything can be 1 over f.
I don't disagree, but certainly neural data, if you record EEG or anything, you plot it out and it looks like 1 over f.
Well, exactly.
Some bumps in it. And I do take the point. So everything, I was having a discussion earlier that over the last couple of days, I can't remember with who, that especially sort of in the early or late 2000s, noughties, whatever, there was this explosion in network theory, and everybody got very interested in scale-free networks, criticality. And you had this sort of tsunami of papers which went, oh, look, we analyzed this network and we found
a small world.
Right? And then you sort of went, yes, and your point is, and but that's all they were saying. So
yeah, we learned that everything is a small world network.
Yeah,
over the space of about 5 to 10 years. Yeah. Raphaël, any thoughts on temporal scales, theoretically or empirically in neuroscience?
Well, I mean, we see scales on multiple levels in neuroscience, right? Like owls can hear things at microseconds level to pinpoint locations, and then you have more of your micro day cycle of days in your neuro and microbiome. I think the digestion that gives kind of slow signals. So you can see that multiple scales affect, well, I think for us it's interesting in the body itself. But coming back to climate, you said you need precision on multiple timescales because from my complexity or nonlinear dynamics, very few knowledge, very little knowledge of nonlinear dynamics. You have this exponent, Lyapunov exponent that describes how precise you need to be in order to do predictions, as in you can do in climate or weather forecast. So I guess it makes sense you need multiple timescales, precision on multiple timescales in order to do more precise forecasts. But I feel like if you're, I don't know, this might be a naive way of saying things, but if you're very precise at a very small time scale, why do you need to be precise at larger time scales, right? I don't know if that makes sense.
Yeah, I think that's right. So in the climate models, weather models, that the measurements have appropriate levels of precision for the level that they're working at. So and the challenge is then to integrate those, like we saw in the keynote talk, these even in the visual cortex. So that's right. But so finding these correct granularities to describe what's going on at each level is important, and then interrelating them, trying to do multi-scale analysis. One thing I want to mention about the chronodes decomposition is that because it's a hierarchical, you get different levels cascaded forward. It's automatic. So you're picking out what to look at these different levels, but you don't have to do it driven by science or intuition. You have an algorithm that will give you a coordinate system like that. And I can imagine examples where maybe some layer there in that hierarchy is changing at a different rate than other layers are not affected by the transformations as frequently, but everything there, it depends on the model of time. So it's not clock time that's important. It'll be whatever the important events are at each level, and their durations may be different. Yeah, their time scales may be different. They might be very big and very small. Some levels changing very slowly. Like if a physical system and you don't do anything, you don't put energy in, it's just not going to change until somebody does that. Right?
So would it be wild, or possibly couldn't happen, to have small things changing slowly and fast things changing quickly, hierarchically?
Potentially that could happen.
Yeah.
I don't see why not, but it's I don't know what in the world is like that. Yeah, large things changing quickly, some things changing slowly.
I think
so. Rocks on Earth.
Plasticity actually would be a good example of that. I mean, slowly changing neural weights, but fast changing neural states. Okay, so one other idea thread to wrap up the panel discussion here is on this topic that we started to get to about coarse graining and granularity. This is sort of for my interest really, but I'm curious what the view of the panel is. So we had two notions of coarse graining that came up today, and in some cases they came up like within one talk. One is essentially downsampling or decimation, and the other is sort of going in the opposite direction, like aggregating, adding at increasingly large clusters, right? So these seem like, in the limit, fundamentally different things. Maybe in practice, often give you the same kind of information. I'll expand on that in one area that I'm familiar with. I mentioned this just now. So when we're analyzing brain activity data, particularly neuroimaging data, to some extent EEG data as well, but structural and functional neuroimaging data, there's the term of art for this is called a parcellation. So a parcellation is essentially a set of labels that say this usually MNI coordinate, or this brain area, this coordinate is part of this brain area according to some atlas or a parcellation, sort of synonymous. And the most useful ones of kind of parcellation sets are actually hierarchical. So we have, say, Yo parcellation or Lausanne parcellation level 1, 2, 3, 4, 5. Level 1 would be something like cuts the brain into 100 regions. Level 2 would be 200, level 3, 400, level 5, 600, etc. Typically, they max out at 1,000. It's sort of diminishing returns in neuroimaging analysis when you take sort of parcels that are more than, say, 2-3 millimeters diameter, which would be, I think, around about what 1,000 lands being. But there's lots of opportunity for either choosing or indeed studying how things look when you cut them in, you sort of chop them in half in terms of their granularity. Well, sorry. Yeah, chop them in half, one of those coarse graining concepts spatially. And this is so it's a very kind of spatial multi-scale thing. It reminded me a lot of the Rubik's Cube, in your example. I was seeing the Rubik's Cube, the way you laid out, I know it's sort of the sequence of dimension eliminations to the solution, but it really did look like a kind of spatial parcellation layout in an abstract space. But this is bread and butter of brain imaging, and that area of systems neuroscience is like, what level of spatial grain do I choose to chunk the brain up into, and what are the implications of that? And is there any interaction between those levels? And that's just within the sort of resolution domain of one modality. And you could think about multiple modalities that fundamentally really do span sort of millimeters to centimeters. So spatial parcellations in that space, spatial multi-scale coarse graining, it's a live topic. It's like a cottage industry, honestly. There's like a new parcellation every 2-3 years. It's quite hard to keep track of. But I think it does speak to something that is quite fundamental in neuroscience, which is that we're not really sure whether to, how to kind of chunk things up. But the approach there is, largely speaking, we have this nested set of subdivisions, and they're more or less equally sized. So it's just more or less like chop it in half, chop it in half, chop it in half, chop it in half, and then choose which one you want to work with. Whereas, with your work with the words, right, temporally, maybe with the cube, not sure, but like they're not necessarily like the next one is half the size of the previous one, right? It's not kind of decimated in that way. So those seem like two ways that you can approach the question practically of doing some spatial or temporal coarse graining that could give very different results in certain scenarios. So I'm interested in thoughts on that.
Yeah, coming back to Rubik's Cube, or I think I didn't show the Rubik's Cube. I showed the 2x2x2 pocket cube, but you could apply you get 5 levels for Rubik's Cube, but you can do the pocket cube in 2 levels, so hierarchically. So it just emerges from the structure of the symmetries. So at the top level, you can say, I don't care what the little stickers say, what colors they show. I just want all the corners to be in the right places. And that's a symmetry group on 8 objects. And that symmetry group appears in the pocket cube. And then hierarchically below that is what's the orientation of each of the little cubies at the corners. And that's a let's see, what's called an elementary abelian group with it's a modulo 3 counter, because you can just rotate. All you can do at a corner is you can change the rotation, and there are 8 corners, but only 7 modulo 3 counters appear in parallel there. It's because the position of the last one is determined by the position of the other 7. So this just emerges out of the math. You get a hierarchy, and it says what's important at the top level is where are the corners, and next level is the orientation of the corners, and that depends hierarchically on where the corners are, right? It just falls out of the algebra, and it is a partition in the group. But if you go to irreversible systems where there's some non-reversibility, you don't get partitions in your coarse graining. You get coverings at potentially at all many levels. Yeah.
Hanna, any thoughts on temporal coarse graining? Do you, for example, like where you're plugging the data into your or you're using the microstate computations, and they've been there's been some downsampling decision made, which is let's call it an even-sized decimation procedure. As opposed to, and then you analyze with the symbolic analysis, so you have these potentially arbitrarily long strings. So you have these two types of potential for constructing some kind of multi-scale data features.
Yeah, there might be potential, but that would be like developing some method on top of that, because currently there is something like what type of event we concentrate on and how much memory we have. And the thing is about this scales, what might be important is that if we computationally can have it on
We lost you for a second there, Hannah, but you're back now.
Sorry, my Zoom is messing around. Yes, so I was telling that particularly for the method, that one additional thing for the importance of the timescales could be that we have the thing of the memory, and if we only have a clock scale and we have about 20 letters repeatedly, if we want some periodicity of this change in this microstates, and we just won't be able to see it at all because of how far we can look, and we will just see the one dominant microstate for a while. But if we want to capture something longer, we'll have to go to another time scale to be able to apply this method in there. So, and that's the thing that mentioned about being able to do something good at the smallest time scale, and then you don't need the others. Well, maybe it depends on how powerful of the methods you have for being able to generalize that.
Will effectively be the temporal autocorrelation length sets a kind of natural time scale. So what Hanna was saying is that if you had too fine a time scale, the microstates are so highly correlated temporally correlated at that time scale that nothing happens, they don't change. On the other hand, if you sample too coarsely in time, like way beyond the autocorrelation length, then you end up sampling kind of white noise. So you need to hit the right kind of spot between those extremes.
True. Although I think there's sort of stuff in EEG that's well below the autocorrelation length that you wouldn't see windowed around that.
You can kind of estimate that just by doing an autoregression and looking at the spectral radius. That gives you that autocorrelation temporal length, roughly.
Okay. Any final thoughts from the panel?
Well, I don't know how relevant that could be, but I know Hupfield in 1996, I think, wrote a paper about sequences of words, which Anna's talk made me thought of, that basically you can disregard the length of syllables. So for example, you have a whole word that's AAA, BB, C, D, and you disregard the length of the syllables to recognize the same word that's spoken more slowly. I don't know how relevant that could be to this kind of sequences of on different timescales here. But he proposed different neural mechanisms to kind of encode those syllable lengths rather than the whole syllable itself, and then just doing a little transformation and then reusing like olfactory computational neuronal like neurons like method from olfactory neurons for recognizing odors, but for word recognition. Yeah, I don't know how relevant that would be.
I think that's nice because that speaks to one of the notions of time that Chrystopher articulated for us is change, events, transitions.
Yeah, exactly. So in linguistics, which I used to study a lot. So you've got whatever language you speak, you've got the phonemes that you're detecting, right? And they change in different contexts. But what doesn't matter so much is, as this direction shows, is that the length, the actual duration doesn't matter so much. It's whether those phonemes are hit in the speech, right? And so it's more of an event-based, event-driven notion of time that's relevant. And if you try to reconcile that with clock time in a signal, you end up doing things like dynamic time warping, but it's because it's fundamentally the events that are important, I think. So it's often reconciling these temporal scales is very important, and there's a lot of challenges in doing it, and it comes up all over the place in complex systems.
Yeah, it's fitting and probably not a coincidence that those symbolic computation tools you're using call them words and letters, right? Yeah, and maybe it is a coincidence, but I think you're exactly right. Like speech recognition in particular, a very good example of a kind of temporally smeared or temporally inhomogeneously decimated or cut up, segmented.
Yeah, but I'd like to appeal for people not slavishly looking at the clock and Newtonian time, because that's not necessarily what's important. It's the events, and so in human lives and stories, right? Actually, a quote from Rhodes is, "Life is boredom interrupted by pain."
All right,
where there's certain significant events that you remember, and then there's the things in between you might not, days go by and you might not have any. There's no difference, right? So it's a change. It's changed the difference that makes a difference. That's important, and whatever scale you're looking at.
Okay, so we had two quotes there to end the workshop, guys. We had difference that makes difference is important, and life is boredom interrupted by pain. And on that note, thank you very much for being with us for this panel for today and for the last 3 days. Let's have a final round of applause for our speakers and panelists today, and we'll have it for the entire speakers group as well.